Book nibbles. Big ideas, small bites. When Rebecca Skloot begins the immortal life of Henrietta Lacks, she immediately draws us into the intersection of science and humanity, where women's cells forever altered medicine, yet her own story remained nearly erased. The book unfolds as both biography and investigative journey, bringing to light the life of Henrietta Lacks, a poor Black woman from Virginia whose cells, taken without her knowledge in 1951, became the foundation for some of the most important medical breakthroughs of the 20th century. Skloot tells the story not simply as a tale of scientific progress, but as a layered human narrative, where issues of race, poverty, ethics, and family intertwine with discovery, innovation, and profit. She reconstructs Henrietta's life, follows her descendants' struggles, and immerses the reader in the murky history of medical research at a time when informed consent was barely an afterthought. Henrietta Lacks was born Loretta Pleasant in 1920 in Roanoke, Virginia, into a family that carried the weight of poverty and segregation. Everyone called her Henny and later Henrietta. When her mother died, Henrietta was sent to live with her grandfather in an old cabin that had once been slave quarters on the plantation where her ancestors had been enslaved. Life was hard and work constant, with tobacco farming a central part of survival. She grew up among cousins and siblings in tight quarters, where family ties ran deep and life followed rhythms that had changed little for generations. As a teenager, Henrietta had a quiet yet commanding presence. She was known for her style, painting her nails red, wearing bright clothes, and carrying herself with dignity. She began a relationship with her cousin, David Day Lacks, which led to children while she was still young. Their life together was marked by the constant struggle of raising a family on limited means. Eventually, Henrietta and Day married, moved to Baltimore, and built a life in Turner Station, a community of Black families who had migrated north for work and opportunity. Baltimore offered jobs in steel mills and shipyards, and though life remained tough, it carried more promise than the sharecropping South. Henrietta was devoted to her 5 children, Lawrence, Sonny, Deborah, Elsie, and Joe. Her second daughter, Elsie, was born with developmental disabilities and was institutionalized in a segregated facility where conditions were later revealed to be inhumane. This loss weighed heavily on Henrietta, but she pressed forward, providing warmth and care for her family as best she could. Skloot paints Henrietta not as a saint, but as a vivid, real woman. She loved dancing, enjoyed her friends, and lived with vitality. In early 1951, Henrietta began experiencing pain and bleeding that was unusual even for someone who had given birth multiple times. She felt a knot in her womb and knew something was wrong. At Johns Hopkins Hospital, the only major hospital in Baltimore that treated Black patients for free, Henrietta saw Dr. Howard Jones, who discovered a hard, shiny tumor on her cervix. The tumor was unlike anything he had seen. It was aggressive, fast-growing, and distinct. A biopsy confirmed cervical cancer. She was treated with the standard therapy of the time, radium placed in packs against the cervix. It was painful, invasive, and often ineffective. What Henrietta did not know was that during her treatments, doctors took samples of her tumor without asking permission. This was common practice at the time, particularly in teaching hospitals where tissue samples were routinely collected and studied. Henrietta simply signed the standard hospital consent form for treatment, which made no mention of research use. The sample from her cervix was passed along to Dr. George Guy, head of tissue culture research at Hopkins, who had long sought to grow human cells outside the body. Up to that point, scientists had tried to culture cells, but they died after a few divisions. For years, Guy had experimented with techniques, seeking the holy grail of an immortal cell line that could be studied indefinitely. Henrietta's cells changed everything. Guy's technician, Mary Kubitschek, placed her tumor cells in culture, and within days they multiplied with astonishing speed. Unlike other samples, Henrietta's cells did not die after a few cycles. They doubled every 20 to 24 hours, spreading out over the culture medium with seemingly limitless energy. Guy realized that he had stumbled upon something extraordinary, the first immortal human cell line. He named them HeLa, derived from the first two letters of Henrietta's first and last names. Meanwhile, Henrietta continued her painful treatments, unaware of what was happening with her cells in the lab. The cancer spread rapidly through her body. She endured radium burns, severe pain, and increasing weakness, but tried to keep her condition from her family for as long as she could. She went back to Hopkins repeatedly, but the treatments offered little relief. In October 1951, at the age of 31, Henrietta Lacks died, leaving behind 5 children, a husband, and a family who struggled to understand what had happened. She was buried in an unmarked grave near her childhood home in Virginia, her family unable to afford a proper marker. Even as her life ended, Henrietta's cells were beginning a life of their own. HeLa cells became the first human cells that could be grown continuously in culture, and they quickly spread from Guy's lab to researchers around the world. Scientists use them to test theories, develop vaccines, study cancer, and experiment with the effects of radiation and toxins. The polio vaccine, one of the most celebrated breakthroughs of the mid-20th century, was tested and refined on HeLa cells. Later, HeLa cells contributed to advances in chemotherapy, gene mapping, in vitro fertilization, and countless other areas of research. They even went into space to study the effects of zero gravity on human biology. By some estimates, the mass of HeLa cells grown in laboratories worldwide would outweigh Henrietta herself by millions of times. But Henrietta's family knew none of this. For decades, they were unaware that part of her lived on in laboratories across the globe. They did not know that her cells had been bought and sold, generating enormous profits for biotech companies, or the vials labeled HeLa were standard tools in research labs. When they began to hear rumors in the 1970s, confusion and suspicion set in. The family wondered whether Henrietta had been experimented on, whether doctors had cloned her, or whether she was somehow still alive in laboratories. Without scientific understanding, they grappled with half-truths and myths, feeling both awe and betrayal. Rebecca Skloot's narrative alternates between the scientific history of HeLa cells and her own effort to earn the trust of the Lacks family. When she first contacted them, they were deeply wary, having been burned by journalists and researchers in the past. Deborah Lacks, Henrietta's daughter, emerges as a central figure in the book. Deborah had been just a toddler when her mother died, and she grew up hungry for knowledge about the woman she barely remembered. Yet she was also protective, fearful of exploitation, and haunted by questions about her sister Elsie's tragic institutionalization. Deborah's journey with Skloot becomes one of both revelation and pain. She learns about her mother's contribution to science, but also uncovers the injustices done to her family. While HeLa cells had generated profits for pharmaceutical companies, the Lacks family remained poor, struggling to afford even basic health care. This irony cut deeply. Henrietta's cells helped develop treatments for diseases, but her descendants could not always see a doctor themselves. Skloot captures the raw emotion of these moments, the mixture of pride in Henrietta's legacy and anger at the inequities that denied the family recognition and benefit. Skloot also explores the broader context of medical ethics in mid-century America. Informed consent was not a standard practice in the 1950s. Patients, particularly poor and minority patients, were vulnerable to having samples taken or being enrolled in studies without clear explanation. The Tuskegee syphilis study, where Black men were left untreated without their knowledge to study the progression of the disease, is a chilling parallel that looms over the Lacks story. Henrietta's case forces readers to confront questions. What do patients own of their bodies once tissues are removed? Who profits from biological materials? What obligations do scientists have to the families whose cells they use? The book carefully shows that the scientists who first worked with HeLa were not villains, but products of their time. George Guy never personally profited from HeLa. In fact, he gave cells away freely to promote research. Yet the lack of consideration for Henrietta and her family illustrates how structural inequities and blind spots can have lasting consequences. Later, as biotech industries emerged, companies did profit from HeLa cells, selling vials and using them in patentable processes. Still, the Lacks family received no share of this wealth, fueling a sense of injustice that persists to this day. Skloot's narrative is as much about her own journey as it is about Henrietta's. Gaining the trust of the Lacks family was not easy. Many members were suspicious of outsiders and fearful of being used again. Deborah, in particular, struggled with health issues, stress, and the weight of carrying her mother's story. Yet, as Skloot accompanied Deborah to archives, laboratories, and Henrietta's childhood home, a bond developed. These chapters bring tenderness and immediacy, showing how storytelling itself can become a form of healing. Among the most poignant sections of the book is Deborah's visit to Crownsville State Hospital, where her sister Elsie had been institutionalized. They discover records and photographs that reveal the harsh, neglectful treatment Elsie endured. The pain of this discovery is searing, yet it also brings Deborah closer to understanding the unspoken traumas that shaped her family. The story of Elsie underscores how race, class, and disability intersected in mid-century America to produce layers of vulnerability. Another powerful theme is the way Henrietta's cells blurred the line between person and object. In laboratories, HeLa cells were tools, standardized, impersonal, reduced to code. For the Lacks family, however, those cells were part of their mother, their grandmother, their bloodline. This clash of perspectives created confusion and fear. Deborah sometimes imagined her mother suffering as cells were bombarded with toxins or sent into orbit. She wrestled with the idea of her mother being alive in some form, scattered in vials around the world. Skloot portrays these struggles not as ignorance, but as the natural result of being excluded from conversations about science that directly involve their family. By the time the book reaches its conclusion, the story of Henrietta Lacks is both tragic and triumphant. Her death at 31 was a loss to her family, a silencing of a vibrant life cut short. Yet her cells became immortal, helping to unlock secrets of biology and medicine that have saved countless lives. The injustice lies not in the use of her cells to advance science, but in the absence of recognition and benefit for her family and the broader failure to protect vulnerable patients from exploitation. Skloot ends with a portrait of Deborah, who passed away before the book was published. Deborah's determination to know her mother and preserve her story is one of the book's most moving threads. Through Deborah's voice, Henrietta is restored to the center of the narrative, not as a footnote to scientific history, but as a woman who lived, loved, and left a family. The book itself becomes part of Henrietta's legacy, ensuring that her name is remembered alongside the countless discoveries made possible by her cells. In the end, the immortal life of Henrietta Lacks is about more than one woman or one cell line. It is about the intersection of science and society, the ways in which progress can obscure the humanity at its core, and the need to reconcile innovation with justice. It is a reminder that behind every vial of cells, every experiment, every headline about discovery, there are human beings with lives and families. Skloot's storytelling bridges the gap between the laboratory and the living room, giving Henrietta back her story and inviting readers to grapple with the profound questions her life and legacy raise. The book's impact lies in its balance, celebrating the enormous good that HeLa cells brought to the world while refusing to overlook the inequities and pain. It is both a scientific adventure and a family saga, a history of medicine, and a call for ethical accountability. Above all, it restores Henrietta Lacks to her rightful place in history, not just as a source of immortal cells, but as a woman whose life mattered, whose descendants still carry her legacy, and whose story challenges us to think differently about science, race, and humanity itself. And that's it. Rebecca Skloot begins the immortal life of Henrietta Lacks. Thanks for listening to Book Nibbles. If you enjoyed this bite-sized book, don't forget to subscribe for more quick, insightful summaries. See you in the next nibble.